We're going to be talking about microscopy and cell structure this week. I'm going to separate this week's lecture into two distinct sections, the lecture part and the lab instruction slash demonstration part. I'm going to talk about the more theoretical aspects and the background you need in the lecture, and then walk you through as much of a hands-on lab as I can walk you through without an actual microscope in front of you for the demonstration part. This week's core concept is that images carried in light waves can be magnified and resolved in more detail by using a two-lens system microscope. Some early microscopes used to be one-lens microscopes, but our modern light microscopes tend to be compound microscopes that have two lenses that work in concert with each other. Using a light microscope, the limits of resolution can be extended 1,000-fold beyond what you can see with the unaided eye. That means that you're able to see things that are 1,000 times smaller than you would be able to see without any magnification through a microscope. This slide shows you all the parts of the microscope that I expect you to know about. In the following slides, I'm going to go over each of those parts. I will name them each for you, and I will tell you what each of them does. When you're looking at a microscope, the thing that it stands on is called the base. Then the solid part that goes around the back is the arm. You use the arm when you carry a microscope. When you carry a microscope, you want to get a hold of it by the arm and put one hand under the base. You should always carry a microscope using two hands. The reason why you hold those two parts is because those are structural parts of the scope that are not mobile, which makes them useful to get a firm grip. Microscopes are heavy and expensive. Be careful when you handle them or carry them. The body of the microscope is the part that connects the ocular piece to the nose piece. The body can usually turn so that the ocular piece can be turned to face different directions. The nose piece holds the objective lenses. I will talk about the objective lenses and the nose piece on the next slide. On this side, you see the mechanical stage labeled. The mechanical stage supports the slide for viewing. Sometimes mechanical stages have different contraptions, attachments, that will hold culture dishes or slides. Our microscopes have a slide holder clip to insert a slide for viewing. To the right, below the stage, are the stage control knobs. One of those knobs moves the stage towards you or away from you, while the other one moves the stage to the right and to the left. This slide shows you a close-up of the slide holder. To insert a slide into a holder, press the black lever on the side, on the left side of the stage, to open the slide holder. When it's fully open, it's ready to accept the slide. Put the slide in there and make sure that it's flush against all the sides. There's a little spring in the lever that will hold it in place. Note that none of the glass edges of the microscope slide should be under the stage clip. If any parts are under the slide, then either the slide holder or the slide itself can break. When you want to use a microscope, the first thing you want to do is to turn it on. There's usually an on-off switch on the base that you can turn on. It turns on the light source in the base. The next thing you want to adjust is the amount of light entering the illuminator and through the specimen. You use the adjustable light dial for that. The illuminator brings light into the condenser. The condenser is a lens system under the stage that focuses the light upward to the specimen. Usually you don't have to muck much with the condenser. The iris diaphragm is used to induce contrast in the specimen. The diaphragm has a rotating wheel or lever to open and close, and it helps the aperture to regulate light. It's found beneath the stage. It's another one of those parts that you usually won't use much. Those are usually set by somebody who knows more about microscopes than we do. Then the light will go through the slide or the object that you're looking at on the stage, and the light will enter the objective lenses on the revolving nose piece. The lens closest to the specimen on the slide provides the initial magnification of the image. On most microscopes that you're working with, you will usually have 3 or 4 objective lenses on the nose piece. In our case, we usually have 4. There's a scanning lens, which is 4x. You recognize it by the red ring at the bottom of the lens. There's a low power lens, 10x magnification. It has a yellow ring near the bottom of the lens. The high power lens has a blue ring, and the oil immersion lens has a white ring. It provides a magnification of 100x. After the light goes through the objective lens, and you can switch those by turning the revolving nose piece to have the objective you want to use closest to your slide. After the light goes through your objective lens, it then enters the ocular lens, which is the lens closest to the eye. The ocular lens is usually 10x, which means that the scanning lens then provides a total magnification of 4x times 10x, which is 40x total, but we'll talk about that in the next slide. The way you focus your specimen is with the coarse and the fine focus adjust on the right hand side of your microscope. Both of them move the the stage up and down and bring your specimen in focus on the slide. The coarse adjustment is only used in the 4x scanning objectives. After you get your slide in focus in the scanning with the scanning lens, and you should always start with a scanning lens when you start with a new slide. After you are done with the 4x lens, you will only use the fine focus adjust. Our microscopes are parfocal. That means that once you have the first lens in place, you can just switch to the next lens, to the next lens on the nose piece, and it should almost be in focus. It should just require a little bit of a fine focus adjust. As I mentioned before, you always want to start with the 4x, and why you should do that will make sense in a couple of slides. To calculate magnification on a compound microscope, you have to multiply the magnification of the ocular lens with the objective lens to get the total magnification. The ocular lens is in the eyepiece. The magnification is usually stamped on the housing, and it's usually 10x. The objective lenses, you usually have 4. The scanning lens usually has a red ring, is 4x. The yellow ring, that's the low power, that's 10x. The blue ring indicates the high power, that's 40x. And the one with the white ring is the oil immersion lens, and it's got 100x magnification. Most microscopes are parfocal, meaning that when the slide is focused with one lens, the objectives can be changed without completely losing focus. Whenever you start on a new slide, start with a 4x lens, and then so use the coarse adjustment at that, and then only use the fine focus adjust dial with the 10x, 40x, and 100x lenses. The lenses are rather expensive, and when you move the slide across the front of the lens, you can actually scratch the lenses. And by using only the fine focus adjust style, you minimize the danger of that. Let's talk about some of the properties of the microscope. We will play with some of these properties of the microscope during the lab this week. So the first thing I want to address is image orientation. The image orientation, when you put an image down, when you put a slide down, the image actually that you see through the ocular lens is not the same that you put down. And that's one of the things you're going to discover in the lab. When you may have used one of those little pinhole cameras before, and the image under image orientation on the slide shows you a little pinhole camera. Basically what happens is the image gets reversed because of how the light rays go through the focus through the pinhole and then project onto the film or onto whatever the other side is. And that's exactly the same thing that happens in the microscope. The other thing I want you to pay attention to is the working distance. The working distance is the distance between the objective and the slide. And the working distance gets smaller the closer you get, the higher the magnification is. Meaning that the higher the magnification is, the closer you get to your slide. Which then means that that's the closer you get to your slide, the more dangerous it is that you could scratch the surface of your lens, which is why you only want to use the fine focus adjust. The light intensity is another characteristic that I want you to pay attention to. Basically what happens is light goes through the slide, it gets deflected. And it gets deflected as it goes through the air. There's one level of deflection, and then there's another level of deflection that goes through the slide, and then so light gets deflected more and more. And because the working distance gets lower, you can't capture as much light with the higher magnifications. So it may happen that when you start out looking at your slide, you may have one light intensity, but then you may have to turn up the light dial before in order to see it at a higher magnification. By and large, it's not usually a problem with students in light intensity. They actually tend to start with too much light. So if you don't see anything when you first start on your slide, turn down the light and see if you see more. The other thing to pay attention to on this slide is this little drop of oil. You may have noticed that the 100x lens, that I called it oil immersion lens. And the oil immersion lens, we're going to use it only when we need it. The oil immersion lens requires a drop of oil underneath because of the deflection of the light. The oil immersion lens is so close to the scope that we don't catch enough light. It gets deflected too far for us to see anything. And the oil has a similar refractive index through glass, so it minimizes the deflection and allows more light to still go through the lens. The next thing I want you to think about is the field of view. When you're as you zooming further and further in, basically that's what you're doing, right? As you're enlarging your image, you're actually zooming further and further in. And so this ability to see more detail, see smaller details, since your total field of view is limited, right? You have a certain amount that you can see at once. The higher your magnification, the smaller your field of view. You can see that sort of at the flower you see on your right. The further you zoom in, the less of the flower you see. The flower on the right also illustrates the idea of the depth of field. Depth of field is the sharpness of the focus from the front to the back of the image. And on the first picture, you see that there's a lot of things that are in focus in that picture. But as you zoom further and further in, there are lots more things that are out of focus. And the same thing happens in a microscope. And we're going to play with that characteristic when we play with the threads and the depth of field property. There is a limit to our ability to what a light microscope can do. We can't make any lenses that are much higher than a 1000x lens because it wouldn't help us anything. Because the limit of the light microscopy is actually determined by the wavelength of the light used in light microscopy. Because of the wavelength of light, the resolving power of a light microscope is at the most 0.4 nanometer. This characteristic is called the resolution. The resolution is the ability of lenses to distinguish 2 points as 2 separate points. And I'm using this picture of the Mona Lisa on the bottom to kind of give you an idea of that. So think in terms of pixels. If you have a pixelated image and you enlarge it, you're only going to see pixels that are larger. You're not going to see more detail. And that's exactly the same thing that happens with a light microscope. So beyond the resolving power, items can only get made bigger, but you can't see more detail between them. The last concept we're going to be addressing is osmosis. Osmosis is related to diffusion. And so you know what diffusion is. Diffusion is the desire of a compound to basically spread out as evenly as possible, as far as possible. And when you do Bio 112 with me, you'll hopefully find that it's amazing what all our bodies do with diffusion and osmosis, mostly diffusion, but osmosis is a very, very powerful force too. And one of the beautiful things about both osmosis and diffusion is it doesn't require any energy. But what osmosis is, it's a diffusion through a semi-permeable membrane. So what you have on this picture is you have a beaker, and on the left you have a concentrated sugar solution with the red molecules being the sugar molecules. On the right you have water molecules. The blue molecules are the water molecules. And in the middle you have a partially permeable membrane, which basically is the same thing that you have with a phospholipid membrane. A phospholipid membrane is permeable to water, but it's not necessarily permeable to salt and sugar. But physics still says I wanna try to distribute everything as evenly as I can possibly do it. So since the sugar can't move and can't work towards that, the water is the thing that has to move. So in diffusion, you still have things moving from a higher concentration to a lower concentration. But because the solute, the sugar molecules, cannot move because they can't go across, the water moves from the higher concentration of water to the lower concentration of water. And of course, where the lower concentration is of water, there's the higher concentration of solutes. And the hardest part that goes along with that is the description of hypotonic, hypertonic, and isotonic, because they describe the concentration of the solutes. But the first thing I want to show you is a little bit what this looks like. So we've got this osmosis thing in the beaker. Let's see what happens if we just let this go. Sometimes it helps to actually see the things move, so I've tried to make a little video. So the thing to keep in mind about osmosis is that there's a net movement of water in the direction of the lower concentration of water, which means it's towards the higher concentration of solutes. And so we're going to try to look at that with cells on the next slide. The phospholipid membrane that surrounds a cell is a semi-permeable membrane. It will allow water through, but it limits the influx and efflux of things like salts and sugars and that kind of stuff. There are three terms we're going to have to concern ourselves with: isotonic, hypertonic, and hypotonic. Isotonic, iso means the same. That means on the inside and on the outside of the cell. And isotonic, hypertonic, and hypertonic are comparison values. So you always have to say it's isotonic compared to something else, or hypertonic or hypotonic compared to something else. So isotonic, it's the same concentration of solutes on the outside as on the inside of the cell. That's usually what we compare these things to. That means there's no net movement of water in any direction. Water goes in and out and just kind of keeps on moving in and out. Which in this case of the red blood cell here means that the red blood cell is perfectly happy, perfectly fine. Now in the hypertonic situation, the concentration of solutes, so the sugar or salt or whatever, is higher on the outside than it is inside of the cell. So what I want you to think about now is, what direction is the water going to move? If you have more sugar on the outside, where's the water going to go? If you guessed that the water will move out of the cell from the inside towards the higher sugar concentration on the outside, you are correct. Now, if this happens to a red blood cell, what happens is the red blood cell shrinks. And that actually is not a very dangerous thing for a red blood cell. It's not very happy in this situation. But animal cells can shrink, and then they can recover from that again. The last situation is the hypotonic situation. In a hypotonic situation, the concentration of solutes on the outside is lower than in the cell. So in this case, what's going to happen? What direction is the water going to move? In this case, the water moves towards the inside of the cell, filling the cell. And animal cells don't really have any defenses against, it doesn't have anything that keeps the cell together if there's too much pressure on the inside. So if you put a red blood cell into a hypotonic solution, the red blood cell will swell up and it will burst. And that will be the end of the red blood cell. Now what we're going to do in the lab, we're going to do an experiment with actually an animal cell, with a plant cell. And plant cells are different. Plant cells actually like the hypertonic medium because it brings water inside of the plant. What plants have an adaptation that is a cell wall, which is a rigid outside wall that keeps them from bursting. This is why you can water plants with just regular water. But it would be very unhealthy to take an IV of just regular water and put that into your veins.